Alright, welcome guys to the first video of our AP Biology Codeon series. Today we're going to be going over a topic that's very important for the AP Biology exam, biochemistry. So you might be asking, why is chemistry important? Isn't this a biology exam after all? Well, it is, but we have to know that many of the cellular processes in our body involve chemical reactions, and this is why we do need to go back and study biochemistry for a bit. So at its basic, again, let's review just the fundamentals of biochemistry. First, with atomic structure. We know that all matter is composed of atoms, protons, neutrons, electrons. And one very important idea that we have to know about atomic structures is ground versus excited state. And this is going to be really important for later processes like photosynthesis, which you're going to learn about, like chlorophyll. So chlorophyll is going to involve this distinction. And something else that's really important is isotopes. Isotopes are very useful, and you can just think of isotopes as atoms that are chemically the same, but they differ in the number of neutrons. So carbon-14 and carbon-12, for example. Carbon-14 is going to have 8 neutrons, while carbon-12 is going to have 6 neutrons. And this is going to be really important because carbon-14 is going to be radioactive, which is really useful when you're doing radiocarbon dating or tracing for metabolic processes, and so on. So again, atomic structure, we have protons positive, electrons negative, and in stable compounds, they're going to be the same. Now, how does this relate to energy? Something really important that we also have to review is bond formation versus bond breakage. So if you see on the right, there's two different molecules. One of them is formed by a covalent bond. The other is formed by an ionic bond. So whenever we form bonds, we're going to essentially create or form energy, right? And when we break bonds, we need energy to rip apart these bonds, right? These sharing of electrons, or in the ionic bond, which we'll now explain about, this involves a transfer of electrons. So on the right we have sodium chloride, NaCl. We know that sodium is going to give up one of its electrons, the outermost shell, to chlorine, so that both will satisfy the octet rule. And then on the right we have a water molecule, and this is going to be a covalent bond, polar covalent bond to be exact, because it's going to be an unequal sharing of electrons, which we'll look at more here. So nonpolar versus polar. So these are both covalent bonds. Nonpolar involves the equal sharing of electrons. So on the right we have one molecule of hydrogen gas, H2, and they're both going to share one pair of electrons. So there's going to be equal sharing. Now on the left, we have, again, our lovely water molecule, but this time it's not going to be equal sharing of electrons. It's going to be, the oxygen is actually going to have 2 pairs of electrons, while each hydrogen is going to have only 1 pair. So again, in that case, it's going to be an unequal sharing of electrons. So far we've discussed a lot about the atomic structure, and we've also emphasized a lot about water, which I'll explain more about in the upcoming slide. So these ideas about the molecular structure and atomic structure is going to be fundamental to your understanding of how cellular processes come to play. Now, before we explain about water in the next slide, let's first review hydrophilic versus hydrophobic. I'm sure that you've come across these terms in your AP Biology class or self-study, but essentially hydrophilic means, as we all know, water-loving. This means that any substance that is hydrophilic, which in nature will be ionic, either ionic molecules or polar molecules, they're going to be attracted to water. On the other hand, we have hydrophobic, which is water-hating, or a molecule that's scared of water, as you see in the diagram on the right. This means that the bonds, which most of the time are nonpolar, is not going to feel any attraction to water, which is polar. And these molecules will often be lipids or, as we just said, nonpolar molecules. And then getting back to water, our lovely friend, our polar molecule that is essential to basically most cellular processes and metabolic reactions. So these are just some key properties of water that are essential, and you should definitely know them. I would recommend you just memorizing the bullet points, and just so that when you actually go to the exam, you'll be ready when you're confronted with a question about water. So the first one, let's break it down. Water is asymmetrical because at the intramolecular level, there's going to be, as we said, an unequal distribution or pool of electrons between the hydrogen atoms and the oxygen atoms. And what this allows is, as we see on the right, for something called hydrogen bonding. Because there's going to be a more buildup of charge on the oxygen and a less buildup of charge on the hydrogen. So there's going to be partial charges on the oxygen and hydrogen atoms. So the hydrogen is going to be partial positive, oxygen partial negative, and thus there's going to be an attraction that we call hydrogen bonding. So the H of the water molecules are going to be attracted to the oxygens of the other water molecules. So that's that for the asymmetrical nature of water. And moving on, we have high specific heat. What this means, first let's define what specific heat is. So it's essentially the amount of heat that's required for a substance to absorb to increase that substance by 1 Celsius. So 1 gram of that substance by 1 Celsius. And water, it turns out, has a high specific heat. And why is this important? Well, in marine life, actually, water is essential, or else marine organisms wouldn't be able to survive. It's because it takes a lot of heat for water to to increase in temperature, and thus, if the water temperature is stable, then marine life can be sustained. So it's really important that water has a high specific heat. Now, water also has a high heat of vaporization. What this means that it takes a lot of heat for water to become a gas from a liquid. So vaporization basically means evaporation. And this is very important because in metabolic processes such as sweating, you need a lot of heat. So your body can actually cool itself down by absorbing that energy that is generated through your body heat to evaporate your sweat. Now, another key idea is that water is a universal solvent. That means that it can readily dissolve essentially all polar and ionic molecules, which we mentioned earlier. And then this last one is extremely important for plant biology. It's the strong cohesion tension of molecules, so the attraction between molecules, and this will involve 3 basic mechanisms. So the first one is transpirational polar cohesion tension. So this is what happens when water travels up a tree from the roots to its leaves without energy, because there's such a strong attraction between the water molecules. Now, this is actually amazing, right? And the second mechanism is capillary action. So combining the forces of cohesion and adhesion, we have capillary action, which will also happen within plants. And then last but not least, we have surface tension. This is what allows insects to walk on water, because there is such attraction between the water molecules that that very light organisms such as water bugs can just directly walk on it. And then one thing that I want to mention before we move on, I know this is a lot, but it's that ice, so the solid form of water, is actually less dense than liquid water. So this is very important because it allows, in the wintertime, the organisms under the sheet of ice that you find on a lake to survive, and then that when spring comes around or when warmer temperatures come around, that's going to cycle basically the nutrients and the oxygen up and then cycle the ice back down. And this is what allows organisms to continue to survive in the winter in many places of the world where ice is prevalent. So moving on, we have also something really important, pH. So I'm sure that you guys have seen litmus paper, and so pH, let's first define what pH is. So the pH is essentially the measure of acidity or alkalinity in the solution. So how acidic or how basic something is. And we measure this using a scale. A scale on the right, which I'm sure many of you guys have seen, that goes from 0 to 14. And as we move right to 14, the substance becomes more basic. As we move to the left, it becomes more acidic. And essentially, something that's really important that you have to remember is that every time you decrease in pH in a value, in one pH value, there is a 10-fold increase in acidity. So there's going to be a 10-fold increase in the concentration of hydronium ions or protons. So essentially, acidity is measured by the concentration of protons or H plus atoms in a solution, while the alkalinity of a solution is measured using the concentration of OH minus or hydroxide ions in a solution. So in most organisms, and especially humans, the pH is stable around somewhere between 7 to 7.4, but let's just stick with 7 for now. And our body is special because it can actually regulate itself through molecules called buffers. You don't have to know this essentially, but a good example could be bicarbonate ions. But just remember that our body regulates its pH using buffers. So the next idea that you have to know is something that's fundamental to organic chemistry, but it's to simplify it, you can just think of this term called isomers as structures that have the same molecular formula but different arrangements of atoms. So this reflects a key idea of AP biology, which is absolutely crucial to your understanding, and that's that structure dictates function, right? And the three types of isomers that the AP biology curriculum teaches is, first, it's structural. So this is just a different arrangement of atoms. So as you can see in the picture on the right, there's 1 propanol and 2 propanol. You don't have to know these terms, but just what you realize is that the hydrogen on the middle carbon attached to the middle upper carbon is replaced by hydroxide OH. And conversely, the OH on the upper compound is replaced by hydrogen. So this is an example of structural isomerism. And then the next type that is really important is cis-trans isomerism. So these are just molecules that differ in spatial arrangements, and they're around double bonds. And then the last one, enantiomers. This is also really important. You don't have to know it in depth, but just know that this means 2 molecules that are mirror images of each other. So we have, for example, one right hand, we call it right hand molecule, and one left hand molecule. So if they look like mirror images, then just know that that's also a type of isomer. All right, now we're going to dive into basically the fun part of biochemistry, and this will cover most of the organic compounds. Actually, 4 organic compounds that are crucial to AP biology, the AP biology curriculum. And the first is carbohydrates. So we know these are found in pastas, breads, any source of quick energy that we so call it. And the The definition of a carbohydrate in simple terms is basically a molecule that consists of carbon atoms, hydrogen atoms, and oxygen atoms. And the generic formula is CnH2O. So there's three main types that you have to be familiar with. The first one is something called a monosaccharide. So these are the monomers, as we see on the right. This is a molecule of glucose, so very sweet. I'm sure that you guys have heard also galactose and fructose, which are also isomers of glucose. Now, when two monosaccharides come together and they form a bond through a process called dehydration synthesis, water is actually removed, and then a bond forms between the two monosaccharides, and this creates a disaccharide. You can also call dehydration synthesis as condensation, just a quick note. And then how do we break apart disaccharides? This is going to be through a process called hydrolysis, which is the addition of water. Now, the last type is also very important because they're going to be polymers that are crucial to supplying life and structure and energy to a lot of organisms. And these are polysaccharides. So polysaccharides are basically polymers of monosaccharides that are formed by condensation reaction. And some quick examples, so two structural polysaccharides in animals, this is chitin. In plants, this is cellulose. And then two examples of energy storage polysaccharides are starch, which is found in plants, and glycogen, which is found in animals. So these four are extremely important for your understanding, and you should definitely memorize them. Moving on, we have the second type of organic compound. We have lipids. So a lipid is essentially any molecule that is composed of a glycerol molecule, as we see in green on the right. So this is going to be a molecule that's going to be polar. It's going to have alcohol OH groups coming off, and then that's going to react with three fatty acids. So we see in red the three fatty acid chains, and this is going to these fatty acids are going to have carboxyl groups at the left side. So you'll see C double bond O. So when the alcohol functional group, which we will explain about more later, reacts with the C double bond O carboxyl group, we form a lipid. And lipids have many functions, such as energy storage, for structure, and also for messaging, for hormone messaging, which we call endocrine processes. So these are just some examples of functions. And one very, very important lipid that you have to know is something called a phospholipid, which I'm sure that you guys have also heard about. This makes up the semi-permeable membrane lipid bilayer found on cells and also organelles. And this is essentially a glycerol, but there's also going to be a phosphate group involved. Next, we have proteins. So proteins are extremely, extremely important. They have a lot of functions. So the main elements that you'll find in proteins are, I have a good, a good way to memorize it. Just remember CHONSP. So carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphate. And we have, my bad. So not phosphate, phosphorus, and we have the building blocks, as we know, as amino acids. These are going to form peptide bonds. So one of the amino groups is going to react with the carboxyl group on proteins, and that's going to form something called a peptide bond, which we know gives rise to proteins. So as we said before, proteins have a lot of functions. These include growth, repair, signaling, regulation, enzymatic activity, and also movement. Something that's really important about proteins is the way that proteins fold. And I I emphasize this because this is extremely important for the AP Biology exam. You have to know these different structures. So at the most basic level, we have primary. We know that this is just the linear sequence of amino acids. This is going to be unique for every protein. And the secondary structure, moving on, is essentially formed when hydrogen bonding occurs between the amino acids. Now, this is not going to be on the R group, which is basically the unique section to each amino acid, but it's going to be through a lot of times the backbone of the polypeptide chains. And then tertiary, on the other hand, is formed when R groups interact. So these long R groups can these interactions give rise to a variety of different bonds interactions, such as hydrogen bonding, ionic bonding, hydrophobic interactions, disulfide bonds and bridges. So just know that what you need to know about tertiary structures is that it forms from R groups. And then quaternary, just a big molecule formed from multi-polypeptide chains. And a lot of factors will affect the way that proteins fold. So these conditions will include anything from pH and salinity to temperature, and these could all result in a process called denaturation, which we know is just basically breaking apart the protein entirely so that it can't function. Now, there's going to be proteins which you might have heard or might not have heard. This is a bit on the more detailed side, but chaperone proteins that are found naturally in organisms are essential to refolding these misfolded proteins. And then the last organic molecule or organic compound that we have are nucleic acids. We know that nucleic acids are formed from monomers called nucleotides. So on the right we see a nucleotide, which is composed of a sugar, in this case it's ribose, so that corresponds to RNA. We have a phosphate group coming off the 5' carbon, and we have the nitrogenous base on the upper right. So these three are essential. We know that the nitrogenous bases that are critical to the formation of these nucleotides are adenine, which bonds to thymine, two hydrogen bonds, and then cytosine to guanine, three bonds. But in the case of RNA, so what I just mentioned was DNA. In RNA, it's going to be adenine and uracil. So that's going to be a difference between RNA and DNA. So RNA uses uracil while DNA uses thymine. And then another difference is obviously the sugar. In RNA, it's going to be ribose, which on the off the 2' carbon, there's not going to be an oxygen on deoxyribose, but there's going to be an oxygen ribose. So I think this picture was actually a mistake. This is supposed to be deoxyribose. And then these are just some basic functional groups that are really good to memorize. We have amino, which is essentially a nitrogen atom with two hydrogen atoms coming off. These are going to be found in proteins. We have a carboxyl group, so C double bond O, and then we have a OH group coming off of it. This is also going to be found, as we said, in proteins. And these two are going to form something that we mentioned earlier called a peptide bond. We have hydroxyl, so this is going to be OH. These are going to be found in carbohydrates. So really important. We also know that glycerol also had it as well. And then last but not least, phosphate. We saw this in nucleotides, really important. So PO43 minus. It's also going to be found in an extremely important molecule called ATP, which provides energy to all living organisms. So that is that. I hope you really enjoyed this video about biochemistry, and it's an overview on how biochemistry is extremely important for the EP Biology exam. If you have any questions, please comment below and like and share this video, and also subscribe for more content. This is Codeon, and we'll see you next time.